I guess we are now talking about parts of the cell wall. And it looks like I got my little microphone going on. All right, so the cell wall of prokaryotes, this is, again, as I just said, one of the most important parts of microbiology, one of the most foundational parts of our discussion, so across this semester. So good morning, and maybe are there any questions before I start talking about this? No? Okay. You guys ready? Okay. All right. Thank you. All right. So the cell wall is a structure that provides strength and rigidity and shape to prokaryotic cells. Yeah. It is a rigid capsule. I don't know if I should use this term. Maybe I should get rid of that term. Surrounding the plasma membrane. We'll talk about the plasma membrane shortly. We don't have cell walls because we're flexible. Right? The fact that we can move is a trade-off for the toughness of our cells, so to speak. It prevents what's called osmotic lysis. Do you guys remember osmosis from your physio classes? We'll discuss it briefly in Chapter 4, Part 2. Yeah, so osmotic lysis is, you understand lysis? What does that mean? Breaking down, what do you think? Sure, lysis is a term, the term lysis is flexible. It can mean cut, it can mean pop, it can mean break, all about the same, yeah? So osmotic, in this case, maybe the best term would be to pop, like a balloon, yeah? Does that make sense? But to say break, to say break down, destroy, those are all something akin to it, yeah? Okay, so it's made out of peptidoglycan insofar as we're talking about the cell walls of bacteria, yeah? Okay, peptidoglycan, as we learned last week, as I recall, yeah? No, wait. There was snow last week. 2 weeks ago, last time we were here, we learned about the basic structure of peptidoglycan. And here it is, just for review. It is a polymer of repeating, alternating glucose-modified subunits. N-acetylglucosamine, which is where we have an amino group, we have an acetate functional group put onto that amino group, and so N-acetylglucosamine is one of them. And then same here, only with this thing here, which is muramic acid, and that's on the other. So alternating nag-nam, 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 and that is at least the carbohydrate portion of peptidoglycan. So it's a pretty complex structure, because along with the carbohydrate-based structure, that picture, right, your textbook tries to coordinate with color, right? We have pink and purple here, and you can see the pink and purple here to show our alternating nag-nam, nag-nam is linked together with side chains and cross bridges, which are basically special amino acids that link together to chains, yeah? And why is that? Well, to provide strength, yeah? To reinforce it. Guys good with that? Questions? Okay. One special thing about these amino acids is that some of these amino acids, as it turns out, are right-handed amino acids, which is very unusual. All your proteins use left-handed amino acids. We talked about that 2 weeks ago. These can be right-handed. None of the amino acids in your bodies are right-handed. They're all left-handed. Okay. Unless you eat bacteria in yogurt or something, yeah? In which case you have them in your stomach until you digest them. Is that good? Okay. So gram-positive. So why do we care so much? Well, because pretty much all of the bacteria we are going to, now that I'm thinking about it, with no exceptions, every single bacteria that we are going to culture on a plate in this class is going to have a peptidoglycan cell wall. Okay? That does not mean that all bacteria have peptidoglycan cell walls, but all gram-positive bacteria and all gram-negative bacteria, and we'll learn the difference between them very shortly, but all those bacteria have peptidoglycan cell walls, and all those bacteria are directly relevant to human health. Yeah? Those are the kinds of bacteria we see often as pathogens and as flora, as resident flora. Yeah? Good? Questions? Okay, so let's look at the differences between gram-positive and gram-negative bacteria. Gram-positive cell walls, we can see, have multiple layers of peptidoglycan. Yeah? We can see in the picture at the bottom right here, 1, 2, 3 layers of peptidoglycan. Again, linked together with side chains and cross bridges to link together the carbohydrates in the individual layers, and then multiple layers to each other. Yeah? Guys with me here? So these blue amino acids, what are they doing? They're linking together the carbohydrates, the NAGs and the NAMs, in one particular layer. What are the green ones doing? Well, it looks like those ones are linking together the individual layers, one upon the other. Yeah? So gram-positive cells have multiple peptidoglycan layers. What's this at the very bottom, this yellow stuff with the purple in it? That's the plasma membrane. Yeah? And the yellow stuff is the phospholipid bilayer. Hopefully you're already familiar with plasma membranes and phospholipid bilayers. If not, we'll be talking about them shortly. Okay, so there are some other things. Teichoic acids are in gram-positive cell walls. They're not in gram-negatives. It's not completely known what these things do, but we do know that gram-positives have them and gram-negatives don't. So when we look at pictures of gram-positive bacteria, this is a picture of this is a light microscope image, yeah? This is a picture from a light microscope. Can you see the gram-positive bacteria? These are the little guys. They're rod-shaped, yeah? They're bacilli, right? This is actually Bacillus anthracis. These blobs here are white blood cells, and this is a blood sample from somebody that's in a world of hurt, yeah? This is anthrax, which is a pretty notorious and nasty pathogen, and if somebody's got all these anthrax bacteria in their blood, then that's trouble. So, on the other side of that, let's compare gram-negative bacterial cell walls. Well, we can see here the gram-negative bacterial cell wall has a single layer of peptidoglycan. Yeah? There are no side chains and cross bridges reinforcing it. See that? And that's because there's only one layer. Yeah? Furthermore, we see 2 phospholipid bilayers in a gram-negative cell. Okay? So these are some key differences between gram-positive and gram-negative bacteria. Yeah? Guys good? Questions? Okay. Gram-negative cell walls, again, a single layer of peptidoglycan, 2 membranes, an outer membrane and a plasma membrane. In between them, just a single layer. The fact that they have 2 membranes creates something called a periplasmic space. And yeah, so here are some pictures of gram-negative rods. We can see the picture here shows these guys as sort of pinkish, yeah? why were the gram-positives purple and the gram-negatives pink? Well, the reason is because that when we do a gram stain, and I think we're doing one today, when we do a gram stain, it's what we call a differential stain, and it has the ability to tell these two kinds of bacteria apart, right? So what we have in the gram stain is something pretty special, something that can highlight differences, fundamental differences between entire groups of bacteria based on adding a few stains, yeah? Guys with me? Questions? And the net result is that when we do a gram stain, gram negatives tend to look kind of pinkish, and gram positives look very dark purple. Yeah? Guys with me? Great. So that's most of it. We'll talk more about the outer membrane, the membranes of both gram positives and gram negatives shortly. But let's review a little bit. So gram positives, they have thick layer of peptidoglycan, multiple layers of it, and just one membrane. Gram negatives have a thin layer of peptidoglycan, an outer membrane along with their inner membrane, and in between them a periplasmic space, a periplasm. Yeah, so there are some special parts about the outer membrane of gram-negative bacteria. They have something called LPS, lipopolysaccharide, and you might put, no, don't stop recording, okay. You might put LPS as a big, as an acronym, because it's an acronym you might end up hearing fairly often in your jobs, in your future jobs when you get them. Okay, so LPS is lipopolysaccharide, and here's a molecular structure of LPS. It might not look that clear to you if you're not so familiar with how to identify molecular structures, but have a look here for a second. Do you see anything you recognize? Do you see, so first of all, let's dissect the word. LPS, lipopolysaccharide. What can we infer from this name? What is this molecule made out of? Many sugars and lipids, yeah? Polysaccharide, many sugars, and lipolipid, yeah? Can you see the lipid part of here? I guess it's fairly easy because it says so, yeah? Down here, and you can see sort of zigzags here. So these zigzags, those are, each little zig and zag is a carbon atom in a carbon bond. You remember looking at lipids and seeing that they are often long chains of what we call fatty acids, yeah? And those are hydrocarbon, yeah? Guys with me here? If you don't remember this, you should double check. You should have that chapter 2, part 2 out there and turn a couple of pages until you see the lipids, and you'll see little zigzags here, right? As it turns out, this part will lodge itself into the phospholipid bilayer because nonpolar things like to hang out with nonpolar things, yeah? On the top, what do these look like? Well, these kinds of these kinds of things, these are all the carbohydrates. We have 2 yellowish carbohydrates, and then we have some green carbohydrates, and then some blue ones, and then some red ones, yeah? But each of these things is essentially a little carbohydrate like glucose-type hexagon, yeah? Now, not every one of these carbohydrates is glucose, but you get the idea. Each one of these is a monosaccharide, and so we have maybe 24 carbohydrates here, 5 here, 4 here, yada, 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 right? Why do we care? Well, this molecule is extremely antigenic. Do you guys know what that means? We talked about it a little bit last week, and it's a technical term that we'll have to talk about in a lot of detail in the future. Yeah. But things that are antigenic are things that they cause an antibody reaction. Yeah, so pretty much that's the hard and fast definition. They make your immune system raise a ruckus, raise a fuss, yeah? And in the case of LPS, they raise such a ruckus that, that your own immune response can be harmful to you. It can cause shock, yeah? And that shock causes rapid loss of blood pressure and so on. It's a toxic shock is quite a life-threatening problem. So so as it turns out, if you have gram-negative bacteria in your blood and they are killed, right? If you kill them through antibiotics or something, your problems are not done yet, because once the bacteria breaks apart, the LPS is released, and your immune system says, "Oh my God, there's a bunch of LPS here," and it responds by initiating a very dramatic response, which includes loss of blood pressure and peril. Okay, so anything that makes a bacterium is a structure of which the bacterium is made. LPS, a gram-negative bacterium, is made partly out of LPS. Yeah, it's fundamental to the cell wall. Can you see where the LPS sits here? It's got the little box here. It's the little purple things, right? This purple with a little string coming out. The little string is what? That's the polysaccharides, and the little purple base part, that's the lipids, yeah, that sticks into the cell membrane really well. Guys with me? Okay. So it's when we look at the structure of the outer membrane of this bacterium, this gram-negative bacterium, it's made out of LPS, yeah? But again, the LPS is a toxin. When that cell dies and the cell falls apart and the little pieces float around your bloodstream, if those pieces include LPS, you're going to have a very dramatic immune response. Is that clear? Okay. All right. Questions? No questions. Okay. So, again, as I said before, the gram stain is able to identify gram-positive versus gram-negative bacteria. Yeah? And we'll talk more about the gram stain itself when we're doing lab today. Okay? Okay. So, why do we care about whether a bacterium is gram-positive or gram-negative? Does the outer layer matter? Clearly, right? If we have a gram-negative, then we have the potential to have LPS, which is strongly antigenic. And we need to be mindful of that. And if we have a gram-positive infection, maybe we don't need to worry about that as much as we do if we have a gram-negative infection. Yeah? So that's one vital reason why having a correct diagnosis of whether you've got a gram-positive or whether you've got a gram-negative matters. Any other reasons? You might already know these reasons, depending on how much background experience you're bringing into the class. Why, and again, I'll re-ask the question, why, besides LPS, would we care if we've got a gram-positive or gram-negative infection? Did you guys hear what, and your name is David, right? Is it really David? You wouldn't pull my chain about that, would you? Okay. All right. Did you guys hear what David said? Did you hear what David said way in the back? Would you belt it out, please?
I was saying I thought it had something to do with the kind of antibiotic that was treated.
You hear that? What do you think about his answer? Absolutely. That is a critical thing, right? Gram negatives are not treated with the same antibiotics that gram-positives are. And that's a generalization, but it's actually, it's fairly robust generalization that gram-positives and gram-negatives are treated differently in terms of the antibiotics that we use to get rid of them. Yeah?
We talked about like the cell wall is harder for like destroy that bacteria because of the lipids and remaining in the cell. Is that the same with the double membrane like gram-negative cells? Are those typical, or are they easier to kind of destroy the plasma membrane versus the wall?
Yeah, so ultimately, did you guys hear what Michael said? Yeah? Did you hear? You guys hear? Would you re-ask your question loud, please?
I was saying was a gram-positive cell wall, would that be harder to destroy than a gram-negative cell with two plasma membranes or two cell membranes?
What do you think? Great question. And so the answer is not so simple. But ultimately, what Michael is getting to in his question is a molecular explanation for why we treat gram-positives and gram-negatives with different antibiotics, okay? And ultimately, it comes down to molecules those bacteria, gram-positives and gram-negatives, are made out of, okay? So let's look at that. First of all, here I have a picture of, so distinctive characteristics of prokaryotic cells make ideal drug targets. And I think we talked about this last or 2 weeks ago, yeah? Do you guys remember this? If you had a pill that you could take, you have an infection. Imagine you have an infection with a gram-positive bacterium. You've got a pill that, you've got two pills, a red pill and a blue pill, yeah? Okay, and the red pill destroys, why don't I make a green pill and a purple pill? Because I don't want to, there's already connotations of red and blue pills from movies and so on. So you got a green pill and a purple pill. One pill, the green pill, destroys all DNA, yeah? The purple pill destroys peptidoglycan, all peptidoglycan. You've got a bad bacterial infection. Which pill do you want to take? What do you think? Raise your hand for a green pill, the one that destroys all DNA. Raise your hand for purple pill. Okay, good, right? Looks like pretty much everybody got it, yeah? So somebody explain why. Why would you want to take the purple pill instead of the green pill? You guys got the right answer, yeah?
We don't want to kill all the DNA. We just want to destroy bacteria.
If you take the green pill, do bacteria have DNA? Yeah, they do. All living things have DNA. And so if you take the green pill, those bacteria, their DNA will get destroyed and they'll die. Good, right? Except there's a little problem, yeah? And what's that? All of our cells have DNA too, right? And so we would expect that our cells would be in danger as much as the other cells. If we knew that the way a particular drug worked was by destroying DNA, we would be very, very careful taking that pill because, and in fact, that seems categorically bad, yeah? Because our cells depend on DNA as much as the bacterial cells depend on DNA, right? Whereas, what about the purple pill? It's going to destroy the peptidoglycan of those bacterial cells, yeah? It makes them vulnerable. Does it make them die? Usually it does, but again, we might think, well, that cell wall is intact, or the cell wall is destroyed, but the cell membrane underneath it might still be intact. But certainly, if that cell wall is providing protection for the cell, right, then then there's so much the less protection. Does that make sense? Right? So, the environment will be the one to deliver the coup de grace to those bacteria. You understand what I mean by that? Right? In other words, taking away their cell wall might not necessarily make them die per se, but it will leave them vulnerable such that the killing blow will happen easily by some bad twist of luck inside your body. Does that make sense? You guys with me? Okay. And is taking something that's going to destroy peptidoglycan going to harm you? Why not? We don't have peptidoglycan. There's no peptidoglycan that our bodies are made out of to destroy. Does that make sense? So I've summarized that right here. Since humans lack peptidoglycan, we should not be harmed by molecules which prevent its assembly. Does that make, do you guys hear that? Okay. And notice that I used a little legalese here. Should not. Is that a guarantee? No, it's not a guarantee, right? There are molecules in our body, and if we have one molecule that interacts with peptidoglycan and destroys it, that molecule might do some other stuff too. And by bad luck, by a fluke of molecular shape interaction, it might mess up some other structure inside our cells that superficially resembles peptidoglycan. Yeah? Does that make sense? So that possibility still exists, but as it turns out, for penicillin, right, that has pretty minimal side effects for such an important drug, don't you think? The trade-off, there might be some side effects. There might be allergies, but allergies are different. That's a different kettle of fish. That's bringing in your immune system. But in terms of side effects of drugs, if it reacts with one, the odds that you're going to have side effects are greatly reduced if you don't have that molecule that it interacts with. Is that clear? All right. So we don't make peptidoglycan, and all eukaryotes don't. Okay, so now let's look at this picture on the right, okay? Do you guys recognize this gray thing? What kind of molecule is it? David. I heard some other people saying it too. It's a protein. Netta, was that you that said that? Okay, good. Good, you guys. How did Netta and David know that that was a protein? The shape. A bunch of squiggles. The squiggles are what? Secondary structure, yeah? The fact that it's a ribbon, that's a sequence of amino acids. The fact that the ribbon is coiled, that's secondary structure. And the coils are folded up, that's tertiary structure. So it must be a protein. Excellent, excellent, yeah? Okay, and now that that's mentioned, hopefully you guys recognize that. Well, this molecule here, the one with the red, blue, and gray balls, with one looks like an orangish yellow ball, that one is not a protein, yeah? As it turns out, the caption that I put here is penicillin blocks the action of enzymes that make peptidoglycan. In other words, this thing here with the red, gray, blue, and yellow balls, what is that? That's penicillin. And this thing here is what? The enzyme that makes peptidoglycan. Does that make sense? Right? So that's a distinction. This is not peptidoglycan. This is an enzyme that makes peptidoglycan. We'll talk about enzymes hopefully today. You guys with me? All right. Great. We've spent a lot of time on this slide. It's time to move on. If you have homework, you ought to hand it in. Okay. Lysozyme is a eukaryotic enzyme that secreted by many kinds of cells, which cleaves peptidoglycan. We find it in egg whites, human tears, saliva, human breast milk, and mucus. Yeah? So this is a particular molecule that chops up peptidoglycan. And who makes this molecule? We do. Our own bodies make it, right? So, what does this mean? Well, it means that humans, like all eukaryotes, have evolved in the presence of bacteria, yeah? You have bacteria in your guts now, yeah? What about your caveman ancestors? Did they have bacteria in their guts? Of course they did, yeah? What about if we went back 100 million years ago to dinosaurs and other animals, right? Did they have bacteria in their guts? What do you think? All of them evolved in the presence of bacteria, right? Is it okay for us to have bacteria in our guts? It definitely is. How about in our bloodstream? No, not so much, yeah? So there are some places where we're happy to have bacteria. Our guts is one of those places. There are other places in an animal's body where we definitely don't want bacteria, right? And one of those is the bloodstream or the eyes, yeah? That's not to say we don't have any in our eyes, but we shouldn't have any in our bloodstream, yeah? So, lysozyme is an enzyme made by many kinds of cells, and its job is to chop up peptidoglycan. Do you see here this big blob here? What kind of structure is this blob? It's a protein, yeah? And it has a complex shape. What's this colorful thing going across the blob? That is peptidoglycan. And basically what it's doing, right, is it's chopping it here. It's chopping it here. Every peptidoglycan molecule, if this guy finds it, it acts like a pair of molecular scissors and chops it up, right? Lysozyme. Does that make sense? Yes, because when a bacterium loses its cell wall, it makes it susceptible to osmotic lysis, as we've talked about, yeah? Guys with me here? Okay. Human tears have it. Human breast milk has it. Saliva has it. Even egg whites have it, yeah? And why do egg whites have it? So that an egg doesn't get infected while the little baby chick is growing, yeah? Does that make sense? Okay. All right. So, some more comparisons, gram-positive to gram-negative. Gram-positive cell wall, disrupted by lysozyme, very sensitive to lysozyme. Gram-negative, not so sensitive. Gram-positive cell wall, sensitive to penicillin. Gram-negative, not so sensitive to penicillin. There are some bacteria that can be destroyed by penicillin, but most are not. Or there are some penicillin derivatives that are lethal to gram-negatives, but most aren't. Their outer membrane has endotoxin. This basal body, do you guys remember where this is from? This is kind of a small, fine point. And if you don't remember this, I wouldn't feel bad. The basal body is what makes the flagellum rotate, right? And it sits in the membrane, right? And because this guy has two membranes, he's got twice as many little basal bodies sitting in those membranes. Does that make sense? Right? Okay. All right, moving on. So, the bacteria we're going to grow in class are gram-positives and gram-negatives, yeah? They are not all bacteria in the phylogenetic tree for bacteria, yeah, in the domain bacteria. There are others, and some that can cause disease, that have neither a gram-positive nor a gram-negative cell wall. And some of these guys can cause disease, right? So, Mycobacterium tuberculosis. Have you guys heard of this one? It causes tuberculosis, right? That's a pretty important disease in humans. hugely important, really. It's a prevalent disease of poverty. It's a lot less of a problem in this country, but it's a huge hassle. You can still get it in this country, and if you do, it's a big, big hassle to get rid of. They don't have peptidoglycan cell walls, or I guess they do, but really what makes them distinctive is that instead of a lot of peptidoglycan, they have a little bit of peptidoglycan, and they have a lot of this stuff called mycolic acid. And that makes their cell walls really waxy, and it makes your immune cells, your white blood cells, makes it really hard for them to get a grip on them, to eat them up, so to speak. Okay. Again, damage to the cell wall. Penicillin inhibits the assembly of cell walls of peptidoglycan. Lysozyme breaks the disaccharide in peptidoglycan. You guys understand what I mean by that, yeah? Right? And then once cells are destroyed, once cell walls are destroyed, we have very vulnerable cells, protoplasts. Okay. So much for the cell wall. I think we'll I'm going to actually stop this thing and try to make these a little more digestible. Stop.